Good afternoon, everyone. Thanks for inviting me as a guest speaker, and thank you for joining the final session of today, also everyone online. We will have quite a technical talk, so I hope you're still up to that. Indeed, a collaborative research project between Enza Zaden and Amphasys. And that's what I want to start talking about. Why did we start this collaboration? Well, there were multiple reasons why we did so, some of which were more technical. We were wondering what else do these populations that we see when we do an Amphasys analysis, what else can they tell us? And can we relate this data to germination data or pollen vigor? And then especially for cryopreserved pollen. As we heard also this morning by Mike and Jan, this is important, and cryopreserved pollen is a quite common practice for breeding companies, especially for the Solanaceae crops. In addition to the more technical aspects, well, there were also more personal reasons. We wanted to understand each other's questions, needs, and future perspectives, which could also be a driver for future collaboration. And while we were performing this research, we thought, well, those results are actually quite interesting for you guys as well, which is why we aimed for publication in a scientific journal. And we realized that. So the publication is published in Nature's Scientific Reports last May. Some of you might have read it, some might have not, but we will dig into it deeper now. So our main aim was understanding scatter plots and pollen morphology. So as you may know, the Solanaceae pollen samples consist of different cell subtypes. Like you see over here, the pictures, it's a very obvious result of when you do a pollen germination of either pepper or tomato. And we were wondering, how do these relate to the pollen populations we see when we do the Amphasys measurement? So how did we analyze that? Well, we included 10 cryopreserved samples of both pepper as well as from tomato. Each sample represents a specific genotype and was collected at one of our production locations across the globe. There was a diversity in the amount of time that they were stored. So this was like from 1 to 2 years, but even up to 7 years. That the batches were stored at minus 80 degrees Celsius. Of each and every sample, we did multiple assays. So we performed the Amphasys measurement by 2 technical reps. We performed the in vitro germination, including 3 technical reps. I guess most of you will understand that. This is the germination on a liquid media. And we analyzed the pollen vigor. And all of these assays were performed at both Enza Zaden in Enkhuizen in the Netherlands, as well as here at Amphasys upstairs in the lab. So, well, before really getting started, we first did a pre-study to create the foundation. Since we were going to perform these assays at 2 totally different locations, we really had to standardize our protocol. So these are some of the aspects that we checked in our pre-study. As mentioned, it was really optimization and standardization of the assay. Some things I want to highlight is the rehydration duration, as well as the assessment of the germination media. Here you see the response of dried tomato pollen to a rehydration treatment. As you may know, or as you can see over here as well, when we have dried pollen of the Solanaceae, they have furrows. So they are able to, once they are dried, to kind of fall into themselves, and they have this kind of coffee bean shape. And when you put them in a high humidity environment or in a liquid, they will start rehydrating, as you can see in the little movie here. Eventually resulting in the rehydrated batch that you see on the other side. Obviously, it makes sense that we need a good rehydration before we can check our pollen quality. That was the main message there. Then in addition, buffer osmolarity. So in our in vitro germinations, we noticed that the buffer osmolarity seems to be directly linked with the germination speed. So here you see the effect of the germination buffer osmolarity on tomato pollen germination rate, as well as on the pollen integrity. So on the left side, you see the low level of sucrose, which is a low osmolarity buffer. And on the right side, we increase the sucrose levels, and you get a very high osmolarity buffer. So we noticed here that when we have a low osmolarity buffer, that we have a very quick germination, but we also have a very high risk of pollen tube bursting. So the pollen tends to take up water so fast that it can't really manage the pollen tube growth. However, when the sucrose levels are very high, the germination is very slow, which is the positive thing is that the pollen is very stable. But obviously, to have a good estimate of our in vitro germination, this has to be like in a perfect balance. Okay, so once we had our pre-study and our standardized protocol, we really get it started. So this is comparing viability to germinability. Every batch that we included, we performed an in vitro germination, as you can see an example here. And we identified multiple pollen subtypes, which are the 5 indicated here. So we have the germinated pollen tubes with a pollen tube that is longer than the diameter of the pollen grain. We have ones that germinated, but which have a very short pollen tube compared to diameter. And then we have pollen grains which were able to rehydrate, so they took up water and they are back in their, let's say, normal morphological shape, but they did not form a pollen tube yet. Then on the right side you can see the coffee bean, so these are like the dried pollen grains that remain in this folded shape. And we have the aberrant small cells that we also saw in the presentations before. So we quantified all of these pollen fractions. Here you see an example of the pepper batches. So on the x-axis, it's 1 to 10, the 10 different samples that we included. And this is the analysis that was done at Enza Zaden. So you can see that we, all fractions are indicated in the different colors. And in black, you see the Amphasys viability percentage. So if we visualize 3 of these fractions in a different way on a dot plot, then this is what you see. So I indicated in green, which is the germinated fraction with the long pollen tubes. In yellow are actually all germinated pollen grains, also including the smaller ones. And the black dots represent, let's say, all mature pollen grains, so all of which have rehydrated. And when we take a look, you can actually see that we have a very clear correlation between the viability and the germinated pollen grains, and that's the correlation is like almost a positive linear correlation when we take a look at all the mature cells. However, this is just an analysis of pepper, and it only includes the results of Enza Zaden. So let's add the results of Amphasys and tomato as well. Then this is what we see. So on the x-axis, you can see the viability obtained by the Amphasys measurement, and on the y-axis, the in vitro germination. And as you can see, we have a very high, very good correlation for both pepper and tomato. Then when we include all mature cells, so in this case we call them like the reconstituted fractions, so all of which have rehydrated, this is the very high positive correlation that we found. So, we knew that the Solanaceae pollen samples consist of different cell subtypes, but this is how it relates to the Amphasys result. So, well, we have the dried, dehydrated pollen grains, and actually the ones that are not able to morphologically reconstitute after rehydration can be considered dead pollen. In addition, we don't see any round-shaped dead pollen grains over here, and that's because we analyze dried, cryopreserved, rehydrated pollen. So these pollen might be present when you analyze fresh samples, but over here we don't see them. Then a question remains, because why do these cells indicated here in yellow, why do those ones not germinate? They did reconstitute morphologically, so yes, they took up water, they're back in shape, but they didn't germinate. Well, we believe multiple factors play a role here, and one of them is obviously our assay design. So, yeah, we perform this germination, and after a certain specific time point, we say, okay, now we stop, and now we start analyzing. Obviously, some pollen grains were able to germinate, perhaps some others needed some more time, or some of them already bursted, and therefore we give them, let's say, a wrong scoring. So, yeah, this is obviously one of the factors. In addition, the environment where the pollen is in, the temperature, but also the buffer ingredients and the osmolarity, as we saw before, they all play a critical role in the amount of germination that you will see. And the third aspect, in our case, those pollen samples, they are a blend or a mix from multiple pollen collections. So there can be some differences in pollen maturity as well. And in addition, we have stored them for different amounts of time, and they all came from production sites and were shipped, let's say, to the Netherlands. So they have some different history, which might play an effect. Then let's add the third dimension, which is pollen figure. So of every in vitro germination that we performed, we measured the pollen tube length, as you can see in the picture here. And yeah, we correlated that with the other quality assessment methods. And that's what you see here. So on the left side, you see the viability on the x-axis, and on the y-axis, the average pollen tube length, which in our case is the representative for the pollen figure. And on the right side, you see the in vitro germinability correlated to the average pollen tube length. Well, obviously not a clear linear correlation as we just saw when we compared germinability with viability, but we do see a slight positive trend. So it seems to be the case that higher quality pollen, as you can see in both viability and in vitro germination, they tend to contain a bit more vigorous pollen, at least a longer pollen tube length. Then, since we performed these tests at both Enza Zaden in Enkhuizen as well as here at Amphasys, we had the possibility to check for reproducibility as well as repeatability. And those results are indicated here. So it's on the left side the comparison of the results at Amphasys and Enza Zaden. And as you can see, the results of the Amphasys viability measurement are very consistent. And also the reproducibility of the in vitro germination assay is quite good. Just for the figure analysis, we noticed that it was very hard to standardize because the results between the two locations differed a lot. And you also can see that when you look at the right side for the technical repeatability. In the figure assay, we also see a huge deviation when we consider replicates. So even at the same location, when we have a repetition, we already have differences there, which are less for in vitro germination, and yeah, there are almost no differences for the replicates when we perform an Amphasys viability measurement. So that's also the point I want to emphasize here. for some techniques, like for example, when using the Amphasys, biology is often more variable than technology. So consider replacing your technical replicates by biological replicates then. This brings us to the conclusions and the takeaway message. So in conclusion, we found a solid link between morphologically distinct cell populations and IFC viability. The viable cells identified by IFC, they correspond to the morphologically reconstituted cells, so the ones that took up water. From these cells, a fraction is able to germinate, while another fraction is not. And this is probably due to the germination assay conditions, as well as some other pollen-related factors. And we found no clear link between pollen vigor and both germinability and viability. Our takeaway message is that both in vitro germination as well as the IFC viability are suitable methods for routine quality analysis of cryopreserved Solanaceae pollen. Obviously, there are differences between the two methods, as also indicated here. So standardization, throughput, investment costs, and reproducibility and repeatability. So it will really depend on the requirements that you have, which method is more applicable or more suitable for you to use. Then I would like to thank Sylvan, Ellen, Tanja, and Marco a lot for their contribution to this project. Would like to thank you all for your attention, and I'm happy to answer any questions.